Welcome to SS Physics and the second class for NEET Biology problem solving classes and approaches. This is Debanjan again, and today we will be covering some basic concepts in genetics, and we'll see how Mendel's laws and the first law of Mendel, the second law of Mendel, third law of Mendel, and the monohybrid and the dihybrid crosses are covered, and questions are asked in certain competitive exams, specifically in AIMS as well as in NEET UG. So today we'll be starting first with genetics. And we'll cover two specific topics today. That is, we'll cover the monohybrid dihybrid crosses and Mendel's laws and how it's verified. We will validate it. And we'll also solve 2-3 questions that have been asked already in certain exams. So, firstly, we all know that Gregor Johann Mendel played an important role in developing this field of genetics, and he did this study using Pisum sativum, which is your garden pea. Right? So, and he made some particular observations, and after making those observations, he did some experiments where he took this plant and he took 7 characters or traits and he crossed the plants as in mated and gave the laws, right? So this is the background, and this is the background that I just gave so as to accustom the students again. Now we'll see how these laws were verified with all these experiments. So monohybrid cross, and let me just explain this. Monohybrid cross basically means we are taking parent plant and considering only one trait in that plant. One particular trait, like maybe the height or color of seed or fruit, etc. Like only one particular trait. And in dihybrid crosses, we take the parent plant and we take 2 traits simultaneously, like height and color of seed together. Height and color of seed together. Right? So we'll see what is phenotype, what is genotype, and the various ratios that he got when he crossed these plants of various characters, the monohybrid and the dihybrid crosses, and we'll explore how questions can be solved based on those. So, suppose we'll deal with monohybrid crosses. So let us, for example, take one particular plant and one particular trait. Let us take height. Now, we must understand that we are talking over here about a diploid organism, right? A diploid organism. That means the total number of chromosomes will be 2n. It will be, so whatever it is, suppose 24 chromosomes are there in that organism and it's a diploid organism. So 2n is equal to 24. So the haploid will be equal to 12. What this essentially means is that for every trait, it is controlled by two, or rather we can say by a pair of factors. Now we'll just see. So that is what Mendel said. He defined the term factors, which we today called as gene or allele, right? So allele is basically a variant gene controlling one particular facet of a factor. Right? Suppose we take height and we say that height is controlled by two alleles. So height is, let us, controlled by two alleles. One is capital T and one is small t. So this is allele 1, this is small t, allele 2. Right? So this forms a pair of factors. These are the individual factors which are now called alleles. Right? Now I'll explain how to do monohybrid cross. So this is the concept of allele. Suppose we say that a tall plant. Now when we are saying a tall plant, this tall is what we are seeing, and this is the phenotype of that plant. Phenotype is what we can see, right? Phenotype is what is expressed, right? And the genetic combination, giving the phenotype is called genotype, right? I'll explain. So I said that phenotype of the plant is tall because this is what we can see, and this tall, this phenotype is being governed by a particular genetic combination. Genetic combination means combination of alleles. Right? And this combination of alleles is your genotype. So we have two more terms over here, phenotype and genotype. So we have phenotype, genotype. So tall is phenotype, it's a qualitative thing. Genotype is genetic combination in very simple terms, and this is a quantitative approach, you can say. Even this can be quantitative by the fact that it can be counted. How many tall plants are there, how many medium height plants are there, how many dwarf plants are there, like that. But in the true essence, the quantitative factor is always the genetic combination. Right? Now, suppose, for example, if we say that we crossed a homozygous tall plant, homozygous tall, right? And whenever we are saying homozygous, this term basically means that the form of the allele that is governing the trait, the form of the allele that is governing the trait, they are the same form of the allele. They are not contrasting form of the alleles. Homozygous means capital T, capital T. That means, suppose if T is a controlling factor for height and it is dominant over small t, then homozygous tall means capital T, capital T. That means the genetic composition, again the genotype, will be capital T, capital T. So if someone says, what is the genotype of a homozygous tall plant? If capital T is dominant over small t, the genotype will be capital T, capital T. Similarly, if someone says heterozygous tall plant, what will be the genotype? Genotype will be capital T, small t, right? Heterozygous, that means there are contrasting pairs of alleles. Right? But since capital T is dominant over small t, the capital T will be expressed. So, capital T is expressed. And when capital T is expressed, the phenotype is tall plant because that's the dominant form. Right? So we have covered what is phenotype, what is genotype, what is homozygous, what is heterozygous. Homozygous, tall, heterozygous, tall. And the third combination will be hetero or homozygous recessive. Now, since capital T is dominant, so whenever capital T will be present in the genetic combination, in the genotype, capital T will always be expressed. Whenever capital T will be present in the genotype, capital T will always be expressed irrespective of small t. So, in this condition, capital T is present, so obviously it will be a tall plant. Capital T, small t is present. Again, since capital T is dominant, it will again be a tall plant because capital T is dominant and capital T is present. So, these two are different genotypes, you see. Capital T is one genotype, capital T, small t is one genotype. but they are essentially expressing the same phenotype because one factor, capital D, is always dominant. It is dominant always. So, this will always be expressing a tall plant. So, what will be the condition? When dwarf plant is produced, what will be the condition when dwarf plant is produced? For the production of a dwarf plant, it will always be small t. So for expression of the recessive form, this is homozygous recessive. Right? Homozygous recessive. So, and the phenotype will be dwarf. So, for a homozygous recessive genotype, the phenotype will be dwarf. When small t is suppressed, expression of small t is suppressed. Expression of small t in presence of capital T. That is why whenever capital T is present, capital T will be expressing itself and tall plant will be produced. Only in homozygous recessive condition, small t allele will be expressed and a dwarf plant will be produced. Right? So this is the background. Okay. Now with this background, we'll see some crosses. Now, suppose I say that a cross is made between homozygous tall and homozygous recessive. Right? A cross is made between this. Right? So homozygous tall will be capital T. Homozygous recessive will be capital T, small t. So this is the genotype. This is your parent. Now, this parent will produce certain gametes. Right? Obviously, any parent will produce gametes for the formation of the zygote. Union of gametes will form the zygote. So the gamete that will be formed from this plant will only be of one type, that is capital T, and the gamete from this homozygous recessive plant will be small t. So this is your gamete produced, and these two gametes when coming together will produce capital T, small t. Right? These two will produce capital T, small t. This is your F1 generation. Right? It's also called the first filial. So the F1 generation will be tall plant but a pure hybrid. Tall plant but a pure hybrid. Why hybrid? Because the genotype consists of contrasting pair of alleles. This is the genotype. Consists of contrasting pair of alleles. Right? Now, what Mendel did is, in the second step, he selfed these plants. What did he do? He selfed these plants. That means he mated 2 plants of the same genotype. He mated 2 pure dihybrids. And this is called selfing. Right? And what did he get? So now we'll draw a Punnett square and we'll see what are the various combinations that he is getting. Okay, I'll just remove this so that we have some more space. So now we have capital T small t into capital T small t. What will be the gametes we produced? Obviously it will be capital T small t from this parent, and from this parent as well it will form capital T and small t. Right? Now, one particular parent is producing 2 types of gametes, capital T and small t, because both the gametes, because it is composed of both the pair of alleles. Right? So, we'll draw a basic matrix which is called the Punnett square, and we'll see what are the various combinations. So, you see, from parent 1 we got 2 types of gametes, capital T and small t. From parent 2 we got 2 types of gametes, capital T and small t. And we mated all those. So these are the 4 possible combinations: capital T, capital T small t, capital T small t, small t small t. This into this, this into this, this into this, and this into this. This, and this. 4 possible combinations. And we'll see what are the various genotypes. So what are the various genotypes? TT is there, TT is there, capital T, capital T, small t, small t, small t. These are the 3 types of genotypes. Right? What are the counts? This is 1, this is 1, and this is 2. We have arrived at 1:2:1 is the ratio, and this is the genotypic ratio. Genotypic ratio of monohybrid cross. It's a very important question again. Often asked, what is the genotypic ratio of a monohybrid cross? And there will be various options given: 1:1, 2:1, 3:1, 1:2:1, something like that. And the correct answer will be 1:2:1 is the genotypic ratio of monohybrid cross. Now, we also have a phenotypic ratio. What is the phenotypic ratio? Now we know phenotype is something that we can see. We don't know the genotype, but we can see the phenotype and we can estimate what can be the genotype. Now, this is tall, which is one phenotype. This is also tall. Why is this also tall? Because capital T is dominant over small t, and this is dwarf. So how many total tall plants are there? 1 plus 2, 3 tall and 1 dwarf. Right? So, what is the phenotypic ratio? So, what is the phenotypic ratio and genotypic ratio of monohybrid cross? The genotypic ratio is 1:2:1. The phenotypic ratio is 3:1. It's a very important question. in your boards plus NEET. Right? We have officially arrived at genotypic and phenotypic ratio of monohybrid cross. We can ask you direct questions as to what is the genotypic ratio for a Mendelian monohybrid cross where there is no linkage. We'll again in the next video maybe we'll cover the concept of linkage. Mendel did not take any of the traits, or rather he was lucky that he did not take traits which were linked to one another, and so he did not get a deviation from his own laws, right? Now, in this entire concept, we have also covered one more, or rather two laws of Mendel. We have covered the law of segregation and the law of dominance. We have already covered this too, and I'll tell you how. So, law of segregation happened when the F1 hybrid was selfed. What happened when the F1 hybrid was selfed? Capital T into capital T small t into capital T small t. These were the two parents. This was selfing being done. This is your F1. And what happened is the gametes, they segregated, right? The gametes, they segregated independently of each other. True? The gametes segregated independent of each other. And they combined. This is random combination. And they combined to bring back the original parental types as well as produce hybrid. So whenever you are doing a cross in which there is segregation of the gametes in such a way that they mate to get back to original parental types, plus hybrid, is law of segregation. Law of segregation states that the gametes will be segregating independent of each other and then recombining to form both parental as well as hybrid types. That is called law of segregation. That is Mendel's first law, you can say. And law of dominance is that in a pure hybrid, there will always be one factor, like ability, which will be dominant over small t, and hence will express itself. So law of dominance, you can say it in reverse as well, that whenever a recessive factor cannot express itself in presence of a dominant factor. So whenever a dominant factor is present in the genotype, the dominant factor will only express itself and will get a phenotype that is correlated with the dominant allele or factor. Okay. Now we'll come to dihybrid crosses. Okay, before we come to dihybrid cross, we'll draw one question, and this question was asked in AIMS, sometime in early 2000 maybe. It says that normal maize has starchy seeds. Okay, which remains smooth and dry. So normal maize has starchy seeds which remains smooth and dry, and a mutant form has sugary seeds which go wrinkled when dry. So we have normal seeds, right? Normal seeds is, suppose, capital S, right? They are starchy and dry. And the mutant form exists mutant, let's say small s, which is sugary and wrinkled. Now, this is what is given. Now they have asked, when crossed with a normal plant, the F1 was produced which had normal seeds. Quite obviously, if you cross a mutant plant with a normal plant, so this is your normal and this is your mutant. So obviously it will be producing, the F1 will be producing a normal plant, but a heterozygous normal plant, a normal heterozygous, right? A normal heterozygous. Now they ask that when this F1 hybrid is selfed, what will be the ratio of sugary seeds and starchy seeds? Ratio of sugary and starchy seeds. Right? So this. This is your normal gamete, this is your mutant gamete, right? This is your normal gamete, this is your mutant gamete. See, so you are crossing, this is your parent one, or rather F1, and this is the another parent. So you will get capital S, capital S, capital S, small s, capital S, small s, and small s, small s. And we know that normal seeds, starchy and dry. So ratio of sugary and starchy seeds. Simple. So this is all the dominant forms, and we know that the dominant form is the normal seeds, which is starchy. So this is starchy. This is also starch. This is also starch. This is sugar, carrying only the mutant form. So we have 3 starchy and 1 sugary. Right? We just did this. This question had already been asked. So this kind of questions can be expected from monohybrid crosses, other than the ratios as well. Right? Now we'll be moving on to the dihybrid crosses.